Hello, my name is Jorge Cantú. I would call myself a Latino man, average size. I'm actually the ambassador, the Usher ambassador from Mexico, so really nice to meet you. And it's my pleasure to present our next speaker, Chief Celeste Wyman. Celeste provides genetic counseling for patients and families with ocular genetic conditions seen at Flown Eye Institute at the University of Rochester Medical Center. Throughout her career, she has guided many people with Usher syndrome through their genetic testing experience. She will speak today about the importance and process of genetic testing for Usher syndrome. So please help me welcome Celeste Wyman.
Okay, let me get myself set up a little bit. Okay, it's so nice to meet all of you. I'm Celeste. I'm a white woman in my 30s. I have straight brown hair. I'm wearing my rainbow plaid dress today. Today is Rochester Pride, for any Rochesterians who may know. And I have gold-rimmed glasses. I'm a genetic counselor at Flown Eye Institute at the University of Rochester. So, I need to figure out how to advance the slide. Is it this? Oh, there it is. Is this it? Yeah. So these questions were requested by the conference organizers. They thought they would be helpful questions for me to address. They are, 1, the importance of knowing your genetic type of Usher syndrome, how to get genetic testing, what kinds of specialists to see, what to do if the results are inconclusive. So to address those questions, a few background points that other speakers have also addressed. But to start at the very beginning, genes are instructions for the body. They're packaged in structures called chromosomes. They're made of DNA. We inherit our genes from our parents, and we pass them to our children. Genes make something called proteins, which have functions in our cells. There's a little picture to the side there that's a cartoon of a cell with the nucleus in the middle, the chromosomes and the DNA inside the nucleus of the cell. For Usher syndrome, genetics background, we each have 2 copies of most genes, 1 copy from each parent. When both copies aren't working properly, this can cause an autosomal recessively inherited condition like Usher syndrome. As someone mentioned earlier, autosomal means that both males and females are affected equally. Usher syndrome is an autosomal recessive condition. That tells us the inheritance pattern. And here's a picture that shows that we typically have 23 pairs of chromosomes. Those numbered 1 through 22 are the autosomes. That's where that word autosomal comes from. And the sex chromosomes, X and Y, that you may have heard of. People assigned male at birth typically have an X and Y chromosome, while those assigned female typically have 2 X chromosomes. You can see here a cartoon with a person and their parents, and the person here inherited 1 copy of each chromosome 1 through 22 from his father, 1 copy of the X chromosome from his mother, and a Y chromosome from his father. Now, this is talking about Usher syndrome more specifically. So we can imagine that these blue and red bars are chromosome number 1. And imagine the USH2A gene is in the middle of it, where the checks and X's are. The child with Usher syndrome type 2 inherited 2 chromosome 1 copies, so has 2 USH2A gene copies. 1 chromosome number 1 from father, 1 chromosome number 1 from mother. Both parents each have 1 normal USH2A copy and 1 USH2A copy with a misspelling in it. The misspelling may be called a mutation or a pathogenic variant. The parents are called Usher syndrome type 2 carriers. They don't have Usher syndrome themselves. The child with Usher syndrome inherited a misspelling from both parents in the USH2A gene. When people with Usher syndrome have two different misspellings, the one from their father is different from the one from their mother, we call this being compound heterozygous. You may have heard the term homozygous in genetics. This is when both parents happen to carry the same exact misspelling. This diagram shows that when two carriers have children together, there are four possibilities. They have a 25% chance that they have a child who is unaffected and not a carrier, a 25% chance of having a child who is a carrier for mother's misspelling, a 25% chance of having a child who is a carrier for father's misspelling, and a 25% chance of having a child with Usher syndrome in both misspellings. These chances are independent of each other. If two carriers have children, they won't necessarily have 4 children with 2 carriers and 1 with Usher. Imagine this 4-sided die if anyone plays Dungeons and Dragons. They call it a D4. It's rolled each time they have children together. If a 2 or 3 is rolled, they have a carrier. If a 1 is rolled, they have a child who is unaffected and not a carrier. If a 4 is rolled, they have a child with Usher syndrome. This also shows that if someone with Usher syndrome has children with someone without Usher syndrome who is also not a carrier, all of their children will be unaffected carriers. And these are additional scenarios. On the left, if two parents with the same genetic type of Usher syndrome have children together, all of their children would have Usher syndrome. On the right, if someone with Usher syndrome has a child with a parent who is a carrier for the same genetic type of Usher syndrome, there is a 50% chance that each child they have would have Usher syndrome. Usher syndrome historically, and as we've talked about earlier, is categorized into 3 classic clinical types based on severity of sensory neural hearing loss and presence or absence of vestibular dysfunction. Sensory neural hearing loss is abbreviated here as SNHL. And retinitis pigmentosa, or RP, is the eye part of Usher syndrome. Some people have RP and normal hearing. This may be called non-syndromic RP. RP is a disease of the light-sensing cells in the retina, which is the tissue at the back of the eye, like the film in a camera. People with RP experience progressive vision loss, typically starting with night and peripheral vision problems, as we know. Around 50% of patients with Usher syndrome have clinical type 2, around 44% with type 1, and around 6% with type 3 or atypical Usher syndrome. Now, we've all heard also about subtypes of Usher syndrome, like type 1B, 2A, 1G. This naming system was generally proposed before we fully discovered the associated genes. So it kind of has made 2 different names to remember, the gene name and the subtype name. We might say MYO7A related Usher type 1 instead of saying Usher type 1B. Now that the genes are identified, we can see a spectrum of clinical severity within genes and within types of Usher syndrome. And again, the clinical types of Usher syndrome correlate to a great extent to which genes are affected, but not perfectly. For example, some with USH2A have been found to have vestibular problems, even though this isn't a classic part of USH2. This is a breakdown of the proportions of the different genetic types of Usher syndrome. Around 39 to 55% of Usher type 1 is due to MYO7A. Sometimes MYO7A related Usher is referred to as Usher syndrome type 1B. And 80% of Usher syndrome type 2 is due to the USH2A gene, sometimes referred to as Usher syndrome type 2A. Some people even have RP from the USH2A gene and don't have hearing loss. They can have non-syndromic RP from the USH2A gene. So with that background done, the importance of knowing your genetic type. Some people with hearing loss and RP may be diagnosed with Usher syndrome, then have genetic testing to find out that their hearing and vision losses could be due to 2 separate causes and not due to Usher syndrome, or due to a different genetic condition. So confirming your diagnosis is one important part of having genetic testing. Next, confirming an autosomal recessive inheritance pattern. Confirming the inheritance pattern is helpful because it can tell you the chances that others in the family, including potential future family members, could also have Usher syndrome. Also, knowing your genetic type can help you to connect with others who have the same genetic type and might have some especially similar symptoms as you. And finally, as extensively discussed this morning, currently there is no FDA-approved treatment or cure for Usher syndrome, but it's a very active area of research, and a lot of research studies, including those that may be researching potential treatments for the RP part of Usher syndrome, will require you to have a genetically confirmed diagnosis. Some research studies have even explored treatments that involve injecting a copy of the gene that isn't working properly into the eyes of someone with Usher syndrome. This is called a gene replacement therapy, as we talked about this morning. And researchers would, of course, need to be sure that they are injecting the gene that isn't working properly. How to get genetic testing. I know many of you have already gone through this process. It's best to meet with a specialist who understands genetics. This could be a genetic counselor, a clinical geneticist, an ophthalmologist or an eye doctor, an otolaryngologist, or an ear, nose, and throat doctor. Genetic counselors, clinical geneticists, ophthalmologists, otolaryngologists, they're all examples of the kinds of healthcare specialists who may understand genetics and be able to facilitate your testing. A genetic counselor and geneticist would reliably have an adequate background in genetics, but for otolaryngologists or ear doctors and ophthalmologists or eye doctors, some may have a deeper understanding than others depending on what kinds of patients they see or if they have a subspecialization. There are also other kinds of healthcare providers that might be confused for ophthalmologists or for otolaryngologists. And geneticists have a medical degree and then specialize in genetics. Genetic counselors have a master's degree in clinical and medical genetics. And there's a group called the National Society of Genetic Counselors that has a tool online that you can use to find a genetic counselor. Now, when you meet with the specialist, you should be prepared to review your family and medical histories in terms of your hearing loss, what age, how severe, what frequencies, is it profound, moderate, severe, etc., what type of hearing loss, conductive, sensory neural. These are questions your specialist will ask. What about your vision loss? What age did you develop it? What symptoms do you have? What eye diagnosis was provided? What about your balance? Are you feeling unstable or dizzy? And your specialist will understand if all details may not be available, but those are kinds of the questions that they will be interested in. There's a picture here that is an example of a family history diagram that we call a pedigree. Your specialist should make one for you before ordering testing. This will help them make sure that Usher syndrome seems to be the right diagnosis and will help them interpret your gene results and implications for your family members in your family tree when your results come back. The circles represent those who are assigned female at birth, the squares represent males. Please note that family ancestry is also documented. Father's side originates in Germany and mother's side in Poland in this example. They both have Ashkenazi Jewish ancestry. Sometimes this information about ancestry can be helpful. Some genes are more likely to cause Usher syndrome in different ancestral groups. But again, whatever you know or don't know about your family is okay, and we often work with people who are adopted or completely disconnected from their family. Next, of course, in order to do a genetic test, they'll need a sample from which they can extract DNA. Samples used for genetic testing could include a cheek swab sample, where a Q-tip is brushed gently along the inside of the cheek, a saliva sample, where you spit into a tube, or a small blood sample, a few teaspoons of blood. A little bit about appropriate genetic tests. If you have a hearing loss and RP, the most appropriate test at that point is probably a multi-gene panel of around 2 to 13 genes that have been associated with Usher syndrome. This would be billed through your health insurance, and typically we get results in about 4 to 6 weeks. Another kind of genetic test you might have is through a research study where there would be no charge to you or your insurance, but there can be a very uncertain turnaround time for results for tests that are done through a research study. Well, the first three types of tests on this slide, which says that maybe some less appropriate genetic tests would be hundreds of genes associated with retinal diseases, hundreds of genes associated with hearing loss, whole exome or whole genome sequencing or chromosomal testing. While the first three are likely to find the cause of your Usher syndrome, they are also likely to give you a lot of noise and unrelated gene variants that can be difficult to interpret. Some large retinal disease panels are available at no cost due to sponsorships from pharmaceutical companies or patient advocacy organizations, but may have certain requirements attached to receiving it via that free sponsorship. If such a test is selected due to concerns about cost, it's best done through a genetics professional familiar with the diseases on the panel who can best make sense of any result that may be returned. Some infants with hearing loss have a broad genetic test of hundreds of genes associated with hearing loss because they don't necessarily know that they have Usher syndrome yet, so they wouldn't do a more targeted test. And then a chromosomal test, such as a karyotype or chromosome microarray, is unlikely to find the cause of your Usher syndrome because of the type of technology and data that those tests yield about our DNA. So one of the questions I was asked to address is what to do if the results are uncertain. There's many ways that your results can be uncertain. So this is just to go through the different types of results we can get. A pathogenic variant is the term we use for a known disease-causing misspelling in a gene. A pathogenic variant is known to disrupt the function of that gene and cause disease. An outdated term for a pathogenic variant might be a mutation. You still may come across that term online or find your specialist using it. As an example here, to describe, or I guess a metaphor for a pathogenic variant, it says the "at" ran away. So that sentence doesn't mean anything. Or if you're trying to type the word color and you write CASDF, that doesn't tell us that you're trying to say color. That's like a pathogenic variant. To have a positive genetic test result for Usher syndrome, the lab should see 2 pathogenic variants are identified in a gene associated with Usher syndrome, and we still may need to test parents to help us clarify how those 2 variants are distributed across the person's 2 copies of the USH2A gene. Here's an image that shows that. So you can see the 2 disease-causing variants in the person with Usher syndrome, but if both of those variants came from mom and that person got a normal copy from dad, that wouldn't be sufficient to cause them to have Usher syndrome. This is what we need to see, that one parent has one of the misspellings and the other parent has the other one. This tells us that the person with Usher has a misspelling on both their maternal and paternal gene copies, leaving them with no functioning copy of USH2A, which is the gene example in this. So there also can be negative genetic tests or a benign variant in a gene. There could be a difference in a gene that's not associated with disease. These aren't typically reported on lab reports. An example would be the cat ran away. That's a sentence that makes sense. Or spelling the word color with the U instead of just COLOR, because that's a correct way to spell color if you use the British spelling. A negative test result would find no variants identified in any of the genes associated with Usher syndrome. Now, uncertain results. We could also have what we call a variant of uncertain significance. There could be a change in a gene for which the association with disease is unclear. Maybe you're saying the car ran away. We're not sure if that's what you're trying to say or not. You could spell the word color with an E instead of an O at the end. When you sound that out, that sounds like color, but are you trying to say cooler? That's an uncertain variant. A person with Usher syndrome can have inconclusive genetic testing in many different ways. Maybe they have 2 of these uncertain variants, 2 changes in the gene that we're not sure what they mean. Maybe they have 1 uncertain variant and 1 pathogenic variant. Maybe they only have 1 uncertain variant, or maybe they only have 1 pathogenic variant, when again, in order to be positive, there needs to be 2 pathogenic variants. What to do if the results are inconclusive? These are some questions you could ask. It becomes even more important to be working with a specialist who understands genetics. If a single pathogenic variant or uncertain variant is found, you should first ask if it is in a gene that has been associated with the clinical type of Usher syndrome you are expected to have. If 2 variants are identified and the gene seems to fit, testing family members, for example, a sibling with Usher or parents, can help clarify how the variants are distributed. You can also look back at the testing done. Were any genes associated with Usher syndrome missed? Did the lab that did the test mention any technical limitations in being able to identify gene variants? Based on this, your provider may recommend another genetic test or an additional Usher gene that may have been missed at another lab, and maybe use a different type of testing technology. There are also research studies that involve genetic testing and might use more robust testing technology. Things to remember if your results are inconclusive. Usher syndrome is a clinical diagnosis. We're still learning about genes and Usher syndrome. Consider participating in a genetic database or registry, of which you may learn about some registries at this conference. Check in with your specialist every 2 to 3 years about your genetic results. So in summary, it's important to know your genetic type of Usher syndrome to confirm your diagnosis. Do you really have Usher syndrome? Confirm that recessive inheritance pattern, what that means for family members. Connect with others who have the same type of Usher syndrome as you. Maximize opportunities to participate in research, some of which being treatment research. How to get genetic testing? Just meet with a specialist who understands genetics. Be prepared to review your family and medical histories and submit a sample for testing. And what to do if the results are inconclusive. Ask questions. It might help to test family members or participating in a database or registry. Check in with your specialist every 2 to 3 years about your results. And that's a picture of my wonderful team that I'm so grateful to work with, and it's been a pleasure to meet you all. Thank you.